In this video we will discuss about the final part of this paper and this figure, that is Granzyme and the CD8 plus T cells affects the cognition function. So we are discussing the role of Granzyme and the CD8 plus T cells from the last few videos. In the last video when we ended, we find out that there are 46 genes that are found to be common between the previously published data about the uh, transcription changes in the overexpressed Granzyme system and the differential expressed genes between the age CD8 plus T cells and the young CD8 plus T cells. Between these two systems, there are 46 genes that are found to be common, and based on that 46 genes and this gene ontology analysis are done, and from that they are able to see that the signaling pathways that are downregulated, they have some common things, and those related to the brain border pathways, and this can be seen on the pathways that are downregulated in this epithelial cell development, heart development, elastic fiber assembly, and also the genes that are downregulated to this blood border pathways. So afterwards they did further uh, deep insight using the cell jet package. From that they are able to see that the genes that are expected to be upregulated and downregulated are found to be in this cell jet analysis. That is, IL-1 and CCL are some of the examples that are upregulated, and this L-selectin related pathways are downregulated. This SELL. This confirms that in the case of this overexpression of Granzyme in the ACE or the CD8 plus T cells, it will cause upregulation of the pathways and the downregulation of pathways in a way that will cause a decrease in the cognition functions. Afterwards, they checked it. One of the proteins, that is PAR1, and whose cleavage is associated with a decrease in the brain functions, brain cognition functions. They are found to be decreased because it is cleaved in the case where Granzyme is overexpressed in the young mice, and they are found to be in normal or higher amount compared to the case where Granzyme is overexpressed. And these things they have quantified in the right hand side of the panel R, where we can see that the PR1 is downregulated when the Granzyme is overexpressed, because it is get cleaved more frequently. The chances of cleaving is more compared to the case where Granzyme is not overexpressed, and this decrease or this more cleavage of this PAR1 is thereby associated with a decreased cognition functions or decreased brain functions. So once validating that Granzyme has a role towards these cognition functions through the through overexpressing of the Granzymes in the young CD8 plus T cells, now they wanted to check this role using the opposite experiment. That is, they take the aged mice, and in that aged mice they induce this anti-granzyme small molecules, which is commercially available or which is used commercially also. So they use that anti-granzyme into the aged mice, and then for the control they inject the saline, and then after 14 days or after a few days they checked 14 days from the initial injections. They again did the RAWM analysis, and in the RAWM analysis they are able to see that in the case where anti-granzyme is used, they are able to see a fewer error compared to the case where saline is used, indicating this approach also indicates that the Granzyme has a role towards these cognition functions, and by inhibiting this Granzyme, one can rescue the weakened brain cognition functions. So this is how they come up to the point that it's the CD8 plus T cells that is causing the overexpression of the Granzyme, and which is associated with a decreased cognition functions, decreased or weakened cognition functions. And by targeting the Granzyme or the CD8 plus T cells, from the previous experiments they have targeted CD8 plus T cells also. So by targeting any one of them, they are able to see a rescue of this weaken brain barrier functions to some extent, as the CD8 plus T cells and Granzyme works in a series manner, not in a parallel manner. So by targeting any one of them will affect the overall impact on the weakened cognition functions and helps in the recovery of these weakened cognition functions. So this is what they finally conclude uh, at the end of this paper, that either CD8 plus T cells or Granzyme, any one of them could be targeted to rescue the brain cognition functions, and these are the one that plays or that has a major contribution towards this weakened brain barrier. That's all for this video. If you need to understand more about these experiments and this paper in detail, you can go through the reference that has been provided in the description. Thank you for your time.